Thank you for having me. I'm very excited to be here. Today, I'm going to take you on a journey beyond Titer and show you how Berkeley Lights allows you to identify top producers with favorable product quality attributes within 5 days of cloning. Berkeley Lights is a public company, and I'm therefore required to show this slide. It also gives me an opportunity to take a deep breath and enjoy this moment. To begin, I'm going to briefly talk to you about the Southern Ring Nebula, which seen from Earth appears somewhat indistinct. However, distinct features begin to appear in this next image, which was taken from the Hubble Space Telescope, which is orbiting about 340 miles above Earth. And last month, on July 11th, NASA released photos showing this nebula in intricate detail. This last photo was captured from the James Webb Space Telescope, which is about 1 million miles from Earth. And upon revealing the Webb images, which provide humanity information on the origins of the known universe, this project was described as one of humanity's greatest engineering achievements. Without space telescopes, our best view of the nebula would have been this view. And the blurred, low-resolution image makes it impossible to identify anything beyond the brightest stars. In the early days of cell line development, there was also a lack of sophisticated imaging. Limiting dilution is purely statistical due to poor imaging capabilities, and due to poor imaging capabilities requires multiple rounds of cloning and human verification. The plates and man-hours required to achieve monoclonality assurance make this a costly approach. The images produced by the Hubble Space Telescope were taken using visible light cameras and provided scientists and humanity additional information on the stars and their physical properties. Similarly, the addition of FACS and single-cell dispensing advanced cell line development by increasing cloning throughput. However, single-cell dispensing technologies don't provide any information on the quality of secreted product, which means you have to carry many clones forward to identify a single clone with the required characteristics. The James Webb Space Telescope was sent into space equipped with near-infrared cameras, which provide scientists the ability to image planets orbiting distant stars. This is possible because near-infrared cameras collect light from dim planets that are in close proximity to incredibly bright stars. These planets would otherwise be impossible to image. Clone selection based on growth and titer alone is no longer sufficient because monoclonal cell lines secreting high titers of product are useless if that product is not manufacturable. To find top clones rapidly, you need a technology that provides monoclonality assurance, titer, and quality assessments simultaneously. The Opto quality assays with Berkeley Lights provide information on product quality within 5 days of cloning. And Berkeley Lights thereby provides one of the most advanced cell line development platforms available. And as the James Webb Space Telescope is providing humanity access to the far reaches of the universe, Berkeley Lights is providing humanity access to the intricate details of functional biology, which I will now show you. I'll first walk you through our technology, and then I'm going to provide data showing how our quality assays allow for down selection. The Opto CLD workflow uses Nanopen Chambers, each with a volume of less than 2 nanoliters, which is close to 100,000 times smaller than the well in a 96 well plate. So here you see a 96 well plate, which has, we'll say, a volume of about 100 microliters. And then we have a well of a 384 well plate shown here, which is about a 40 microliter volume. Our nanopens have a volume as depicted to scale here of less than 2 nanoliters or 0.002 microliters. This means that single cells are isolated at what is, from a volume standpoint, equivalent to typical bioreactor seeding density. The small dimensions of the nanopen reduce cell culture and analysis down to the microscale. This allows you to collect relevant information about a clone, such as growth, productivity, and quality, after just a few generations. Optoselect chips are the core of our technology. Cloning, culture, assays, and recovery all happen on Optoselect chips, which contain thousands of Nanopen chambers, as is seen in this depiction here, and a continuous fluidic channel. So reagents are brought in from the inlet of our chip, they flow across the chip, and then exit at the outlet of the chip. There are two distinct regions on the chip. There are the Nanopen chambers themselves, where individual cells are isolated, monitored during culture, and assayed using high throughput automated assays. And then you have the channel above the nanopens, which allows for importing of media, cells, and reagents. At the start of the Opto CLD workflow, I'm going to let you watch the video real quick. So at the start of the Opto CLD workflow, cells from a transfected pool are imported into the channel of the chip. They are then imaged. Once they're distributed in the channel above the nanopens, they are imaged using bright field and fluorescent imaging. The bright field image allows you to identify the location of cells, calculate nearest neighbor parameters, and identify individual cells using those values. Predefined gating schemes are then used to target a specific cell population. So in this case, you're seeing cells that were stained with an Xn5 for viability. You also have a cell surface marker in the FITC channel, and that allows you to identify clones that are viable and secreting high levels or expressing high levels of the target at the surface. The cells are then penned according to predefined gating schemes, and that's what you're seeing here. It's structured visible light is used. Once you've identified targets, we use structured visible light, which you're seeing here, to move the cells into individual pens and clone those cells. Once the cells have been cloned, I wanted to make sure the video is going. Once the cells have been cloned into the Nanopen chambers, the Opto CLD workflow automatically initiates culture and time-lapse imaging. And what you're seeing here is the pens over time using our time-lapse imaging capability. During culture, there's fresh media supplied to the chip using perfusion culture. And so that's just a continuous supply of fresh media that's pushed across the chip. And at this time, nutrients are diffusing into the pen from the channel, waste products are diffusing out of the pen into the channel, and the cells expand. Time-lapse imaging enables you to measure growth rates and doubling time during the culture process, and this is directly followed by assays. Fluorescent on-chip assays are now performed once you've cultured the cells for a number of days. Assays are run in parallel across thousands of Nanopen chambers. So they can be run across a single chip or across up to 4 chips. Each Nanopen is then assigned an assay score at the end of the assay, and these scores allow for ranking based on productivity. In these images, I'm showing an empty pen on the left here, and then increasing intensity in the next 3 pens, which correlates directly with protein production. After about 5 days of culture, where you've cultured the cells and you've assayed these cells, you can select specific nanopens for recovery. OEP is used to direct a clonal colony out of the pen, and that's what you're seeing here. Again, this is structured visible light moving a very specific population of cells within a single pen into the channel. The channel is then flushed with fresh media, and that population of cells is deposited into a single well of a 96 well plate for scale up. Additionally, in this process, there are alternating in-process controls where we actually export a package of media to confirm monoclonality assurance, and this contributes to our monoclonality assurance package. Using this technology, we can provide speed, monoclonality, and top clone selection in a single workflow. As I move forward, I'm going to describe each of the capabilities listed here. And I just wanted to point out that this schematic is showing that we can reduce the number of clones that are moved forward and increase confidence in those clones by doing this growth, titer, and quality assessment very early on in the cell line development workflow. At the beginning, which I showed earlier during that cloning process, we can use selective cell cloning to clone a pool of cells 2.5 weeks post-transfection. And that's what you're seeing here. What we do is there's a transfection that happens, and then 2.5 weeks post-transfection, we can actually move directly into cloning. And that's by using selective cell cloning capabilities, where you're actually identifying the viable cells in a heterogeneous mixture. This allows us to significantly reduce the cell line development timeline. And if you go from this transfection, the timeline between transfection and expansion, you can reduce that from 10 to 14 weeks down to 3 weeks, which is a significant reduction. I've listed a publication here, and if you're interested, you can read this publication, and it will explain this process in detail. To assess pool, so selective cell cloning can be used to reduce that timeline. It can also be used to assess pool diversity. And so you can screen using selective cell cloning. You can screen up to 100,000 clones or cells ahead of single cell cloning. And this allows you to access a pool heterogeneity that you would otherwise lose if you waited for an extended period of time before single cell cloning. In this data on the right, again, I'm listing a publication. It was the same publication as the previous page, where they demonstrated that they could maintain performance when they cloned a pool of cells that had 30% viability. And actually, I say maintained, but they were actually using, compared to a standard workflow using the beacon plus selective cell cloning, they were actually able to achieve a higher normalized titer, which was incredibly impressive. And again, please check out the publication if you're interested. The Opto CLD workflow can also be used to increase cloning efficiency. And in this case, cloning efficiency is the percentage of wells that have expanded once recovery from the beacon, after recovering from the beacon. And compared to FACS, our cloning efficiency is 5 times greater than FACS and 10 times greater than limiting dilution. So we really have demonstrated, and this is in another publication, which I'm listing at the bottom of the page here, we really have been able to increase the overall cloning efficiency compared to traditional workflows. The Amgen team performed a validation study, and it's actually, the publication is listed here, where they demonstrated that the overall monoclonality for the beacon is greater than 99%, and that's compared to 98% for FACS and 96% for limiting dilution. For regulatory submissions, the Beacon workflow also provides you a data package where you're actually seeing the pen, a specific nanopen at the beginning of the workflow before a cell has been cloned into it, the nanopen after a cell has been cloned, and then that nanopen during the culture period, as well as assays. We also provide images of the export and recovery process, and that can be used in your IND submissions. A customer of ours submitted an IND to the FDA, and that was accepted. We have an application note available on our website if you would like more information on that. We were very excited about that. Other customers have also filed. I can't share a ton of details because a lot of it is proprietary, but customers are filing INDs, and this workflow provides a very, very high monoclonality assurance. Regarding titer, Catalent ran a study. Catalent is a customer of ours, and they ran a study, and they were able to demonstrate that they could increase their overall titer compared with the beacon compared to a traditional workflow by 3-fold. The beacon's ability to screen a clonal colony of cells, so the ability to screen a group of cells that have expanded over time, allowed Catalent to gain a better estimate of true productivity prior to scaling up for production. And they were able to reduce their timelines while doing this. All of this is explained in this article called Achieving Unique Synergies in Antibody Expression. Similar results have been demonstrated by more than by many customers. Catalent, I can't list them all, some of it's proprietary, but we have many customers that have demonstrated significant increases in titer using this workflow. Now I want to talk about our quality assays, and I'm going to spend the rest of the talk really introducing our quality assays to you. The reason that quality is important is because antibody therapeutics are becoming increasingly complex, and clone selection based on growth and titer alone is no longer sufficient. Our quality assays put quality first to increase confidence in the clones that you move forward, and this has implications for drug manufacturability, shelf life, and patient safety. The first in our series, our quality assays are referred to as the OptoAssure assays, and the first in the series of OptoAssure assays is the new fully automated on-chip aggregation assay. In this case, fluorescent reagents are used to monitor for protein production. And in a case where you're seeing low aggregation, such as this pen here, you're going to see a diffuse intensity signal. However, if product is aggregating, if the secreted product is aggregating, you're going to end up seeing punctate or spots within that nanopen. Our OptoAssure aggregation assay actually ranks pens according to the number of spots. And if you have a high number of spots, that score is going to be, you're going to get a high aggregation score. And if you have a low number of spots, you're going to get a low aggregation score. We're actually looking for pens that have a low aggregation score. And so I'm just showing data here it's scale-up data that was performed in collaboration with Daiichi Sankyo. And what you're seeing here is that clones have been sorted by their OptoAssure aggregation scores. And this is a heat map. Lighter color corresponds to more desirable attributes at the shake flask stage, such as high purification yields and low aggregation and low aggregate concentration. When assessed over multiple criteria, clones with higher OptoAssure aggregation scores were linked to suboptimal performance. So the higher the score, the poorer the performance. And a lower aggregation assay score is predictive of enhanced performance. Scale-up analysis, again, was performed in collaboration with Daiichi Sankyo. We now also allow, so in the past, our workflows, we provided fluorescently labeled detection reagents where you could monitor protein secretion, antibody secretion, so traditional antibodies as well as modified antibodies. We now are introducing our custom productivity assays, which allow you to use your own product-specific binding reagent. And these are robust for a wide range of secreted proteins. So you can use a fluorescently labeled monoclonal antibody, a fluorescently labeled Fab, or a fluorescently labeled peptide. And these detection reagents can bind a wide variety of secreted products, including BiTEs, fusion proteins, bispecific antibodies, which I'll talk about here in a minute, as well as protein vaccines. And in a recent publication, the University of Queensland demonstrated that they could use this custom productivity assay to screen cells secreting a protein vaccine. This was a molecular clamp vaccine that had a molecular weight of about 600 kilodaltons. So it was an incredibly large secreted protein and relatively complex. And they were able to use the custom productivity assay to identify clones that were secreting 10 times higher titer within 2 weeks of initiating this project. This allowed them to move a vaccine into the clinical trials very, very quickly. And this was during the COVID outbreak, of course. So I now want to talk to you about bispecific screening specifically. And a huge challenge, as others have discussed, a huge challenge in bispecific manufacturing is the fact that while you are targeting this desired molecule, in many cases you end up with undesirable fragments, such as the ones shown here, monomers or fragments themselves. A high-quality bispecific heterodimer is going to bind equal levels of antigen 1 and antigen 2. And to identify clones that are secreting a high percentage of heterodimer, you can run sequential custom productivity assays, such as these shown here. The first custom productivity assay was against antigen 1, and the second against antigen 2. You can then plot the scores to generate a calibrated pen score. We convert the arbitrary units of intensity into a nanomolar concentration, which allows you to compare the two proteins against each other. And in this case, what you're seeing is that a pen that has low calibrated pen scores for both assays is going to appear down here near the origin. And a pen that contains high calibration scores or high intensity for both assays is going to appear in the upper right corner. You can then identify specific, so in this case, multiple mini pools were cloned onto the beacon, and a bispecific antibody, they were all secreting a bispecific antibody. In the first example, the pool that falls along the y-axis, you're secreting high levels of antigen 1 and low levels of antigen 2. And for this second population, you're actually secreting a low levels of that heterodimer, and you're seeing more binding, a 2-to-1 ratio between antigen 2 and antigen 1. To identify clones that are secreting a high level of heterodimer, we look for clones that fall along the line with a slope of 1, which is shown in blue here. Top producing clones that fall along the line with a slope of 1 can then be recovered and scaled and scaled up. At which point HPLC analysis can be used to verify that these clones are indeed secreting a high percentage of heterodimer. And so now let's go beyond titer with this exciting example from a customer collaboration. In this case, 13 clones were scaled up. Clones 1 through 6, which fall along the line with a slope of 1, were expected to have a high percentage heterodimer, while clones 7 through 13 were recovered as controls, and they were expected to have a low percentage of heterodimer. And HPLC analysis verifies that clones 1 through 6 do indeed secrete greater than 90% heterodimer. So that's shown in green here. While clones 7 through 9 secreted less than 70% heterodimer, and clones 10 through 13 secreted less than 50% heterodimer. If you plot the calibrated pen score ratio against the percent heterodimer, you get an R-squared of 0.99, indicating that this is indeed a very effective method for identifying clones that are secreting high percentages of heterodimer. So welcome to the intricate details, where you can identify clones secreting the highest percentage of heterodimer within 5 days of cloning. This workflow allows you to reduce the number of clones that you move forward by screening much earlier in the process, and increases confidence in those clones very early in the process so that you know those clones that you move forward are secreting product that you're interested in. And this capability has been described by a cell line development customer of Berkeley Lights as game-changing for this workflow. So please come visit us at booth 413, and we would be very excited to talk to you about this workflow and about your projects. Thank you for your time, and I'll take any questions.